Hello everyone, and welcome to this video podcast for evolution and diversity. In this podcast, we're going to talk about the overall topic of the origin of life. We're going to begin with talking about the theory of chemical evolution, and then we'll talk about RNA as the first self-replicating system, and then we'll end by talking about the first cells. Before we get started with the first objective of chemical evolution, I want to spend some time going over a brief timeline of life on Earth. I'm going to post this link for you on Blackboard under the same place where you find the videos, so you can go over it if you'd like at some point. As we go through it, I'm going to let you know the main points you need to know for quiz or exam purposes. So as we go through this, let's start with the formation of Earth. And we know that the Earth is over 4.5 billion years old. So up here, this is shown in millions of years, so this is 4.5 billion. So that's when the Earth formed. As you go through this on your own, if you choose to, when you click on these different periods here, it'll give you a little bit more information about those topics. This is one thing I would like you to know. I would like you to know that the Earth is at least 4.5 billion years old. Next, we come along here. At about 3.8 billion years ago, there's chemical evidence for life. Then around 3.5 billion years ago, there's visible evidence for life. And the way they were able to determine that is that there's these structures that we see today, and you can kind of see them here in this picture. They're called stromatolites. And what these are mounds of mineral sediments and organic material. And they are produced by microbial communities. So from that piece of evidence, we can reasonably predict that bacteria and archaea that we see down here existed about that time. So I would like you to know that between 3.5 and 3.8 billion years ago, if you take these two together, that we have evidence of life on Earth. At about 2.7 billion years ago, we see biomarkers. And by that, what I mean is within rocks, we have discovered cholesterol. And cholesterol is made within cells. And so it's evidence that lipids were present. Proteins and DNA, they wouldn't have lasted this long. They're not quite as stable. But macromolecules like cholesterol, which is a type of lipid, are much more stable, and they are actually have been preserved in rocks for 2.7 billion years. You do not need to remember this date. Now, between 2.4 and 2.1 billion years ago, we see what is known as the Great Oxygenation Event. And this is a time period, as the name implies, when oxygen levels started to increase at a very high level. And that's a very important time period because that is what led to the arrival of eukaryotes. So between 2 and 2.4 billion years ago, that's when we see evidence of eukaryotes. And that's another one that you should know, that between 2 and 2.4 billion years ago, eukaryotes arose. So in addition to knowing the time period where eukaryotes arose, I also would like you to know that what set eukaryotes apart from bacteria and archaea is that they contained membrane-bound organelles within their cells. And the organelle within the early eukaryotes that allowed it to survive in such high concentrations of oxygen was the mitochondria. So certainly some important organelles early on would have been the nucleus and the mitochondria, because the mitochondria allowed it to survive in these rising concentrations of oxygen. We'll talk more about the formation of mitochondria in eukaryotes later, but for now I just want to let you know that mitochondria used to be a bacteria, and a primitive eukaryote that did not have a mitochondria and could not metabolize oxygen engulfed that bacteria. And that bacteria, over time, lost some of its functions as a bacteria, but retained some of the functions necessary to metabolize oxygen. And so it became the mitochondria. That is a process, that idea is now known as the endosymbiotic theory. Okay, let's move on. About 1.8 billion years ago, we've found evidence of a eukaryotic fossil in the form of what looks like it's an algae. You don't need to know that date or remember that date. About 1.4 to 1.6 billion years ago, chloroplasts arose. And they formed in this manner very similar to mitochondria, also part of the endosymbiotic theory, where a eukaryotic cell that already possessed the mitochondria engulfed another bacteria that could undergo photosynthesis. And over time, that bacteria again, lost some of its bacterial functions, but retained the photosynthetic metabolism functions. And ultimately, it became a chloroplast. This is also an important date. So I would like you to know between 1.4 and 1.6 billion years ago, chloroplasts were formed. Now I'm going to jump to this one here, origin of animals, and they arose about 635 million years ago. That's also an important time to remember, 635 million years ago. The first vertebrates formed about 535 million years ago. And this is a fossil. It's about 2-3 centimeters in length. It's a jawless fish, but it did have a brain. So it had that going for it, I guess. Next, I'd like to jump over here to early land plants. And they arose about 425 million years ago. And that would be a timeline that I would like you to know as well, 425 million years ago. Then about 365 million years ago, tetrapods from the seas started to invade the lands. And so they had 4 limbs, and eventually those would become the land-dwelling animals. Then what we saw about 340 million years ago is a huge diversification of land plants. 280 million years ago, we see our first early mammal-like reptile. 252 million years ago, Earth experienced one of its 5 mass extinctions. Now, you don't need to remember this name of this extinction or the timeline, but I would like you to remember that on Earth there have been 5 mass extinctions. We often just think about the dinosaur mass extinction, but there were 4 others as well. In fact, this one here we predict was the most devastating, where we predict we lost 96% of all species. So essentially from this, I really want you to remember that there have been 5 mass extinctions on Earth. And we're actually living in the 6th mass extinction event. So then about 155 million years ago was the age of dinosaurs, which I'm sure you're all familiar with. Then about 6 million years ago, there is evidence of a common ancestor that branched off to form chimpanzees in one branch and humans in the other branch. So humans and chimpanzees are connected by this one common ancestor. So from this one common ancestor, one of those branches, as I said, became humans. That is the modern day Homo sapien. And Homo sapiens first appeared around 200,000 years ago. So while we think we're quite important on this earth, and for this time being, I guess we are, we've really only occupied this planet for a very small fraction of time that this planet has existed. 200,000 years over a period of 4.5 billion years. And if you think about just the time that life has been on Earth, so for about the past 3.5 to 3.8 billion years, we've only been here, that is humans have only been here for 200,000 years. And so we just occupy the planet for this small part of time. There's probably a pretty good chance that at some point Homo sapiens will no longer exist and the planet will be occupied by newly derived species. We won't know, and we won't be here. So I guess we can just think about it and imagine what it will be like. Now, I also want to say that this here is just a very brief timeline. I obviously left out many important events, the rise and fall of many different species and genre throughout Earth's life history. Now I'd like to jump into our first objective of chemical evolution. So let me move back to the whiteboard. So let's talk about the objective for chemical evolution. Remember that all life is made up of chemicals. And we talked about how simple chemicals can build larger chemicals, larger macromolecules, and how that ultimately can lead to life. But how did all that start, so to speak? A scientist by the name of Alexander Oparin in 1924 proposed the hypothesis of chemical evolution. And within this hypothesis, this is what was proposed, that there were simple molecules in the ocean. And then there's some kind of input of energy. And then you get some kind of chemical reaction that produces organic molecules, or organic monomers, we could say. These would then combine to form larger polymers, like proteins and nucleic acids and carbohydrates, would eventually lead to a molecule that could replicate itself. And remember, replicating oneself is a characteristic of life. At some point, these larger molecules would become surrounded by a membrane. It would become metabolically active. And at this point, we have life. And at this point, chemical evolution is essentially replaced by biological, or you might say natural selection, based evolution. So thinking about chemical evolution, how might that have happened on Earth? And so there were two models that were proposed. The first is called the prebiotic soup model. And essentially what this model proposes is that in the ocean and the atmosphere at early Earth, there were simple molecules like methane, hydrogen, ammonia. Then through some sort of energy, maybe just light energy from the sun, these molecules in the atmosphere and ocean became more complex, like something like hydrogen cyanide or formaldehyde. These are important ones that could have formed because they're quite reactive. And then maybe with some more energy, and maybe this is coming from heat or maybe even lightning, maybe that led to even more complex molecules in the atmosphere and ocean, and that might have even led to things like macromolecules that would have settled in the ocean, and from there, ultimately, life could have formed. One of the problems with this model is the ocean. The ocean, we know, is very, very big, and so there's going to be a concentration problem. Would you be able to increase the concentration of these chemicals and these chemicals to form these chemicals, and then for these macromolecules to form something that could become life? It's hard to imagine that would be possible. So that's one of the drawbacks to this prebiotic soup model. So let's talk about this second model, and it's called the surface metabolism model. And it's not that only one of these models could be true. It's possible there's a combination of these models. But what we want to think about here with this model is, think about at the very bottom of the ocean, where we have these hydrothermal vents. And it's possible that these simple molecules, like ammonia, hydrogen, and methane, could have went down these hydrothermal vents. You would then still have the same steps where you would form things, as I said on the previous one, like formaldehyde or cyanide or acetic acid. A lot of different chemicals could form then, and then those would then become macromolecules. So that's all the same. But why is this more likely? Well, it's more likely because of two reasons. Within these vents, and let's just draw them inside of here, and I'm just going to draw them like this, they're smaller. So it's easier to have a higher concentration, and this is my abbreviation for concentration, of molecules. This high concentration of molecules increases the reaction to form more complex molecules. In addition, these hydrothermal vents are very hot, right? So there's a lot of heat there. So between the increased concentration and the heat, it seems that this might have worked a little bit better, going from simple to complex to more complex molecules. So remember, these models are just models. We don't know for sure which one is the most accurate. We don't even know if one or the other is the way it actually happened. It could be a little bit of both. We just don't know. But this is all kind of guesswork, because how do we know that early Earth would have had the conditions to go from simple to complex to more complex, regardless if it happened in the ocean or if it happened in a hydrothermal vent? And so this really just remained a untested idea until a graduate student by the name of Stanley Miller in 1953 tested whether or not you could go from a simple to complex to more complex molecule. And so let's talk about that on the next whiteboard. It wasn't until 1953 when a graduate student by the name of Stanley Miller tested this hypothesis. And this test led to more tests, and eventually the idea of chemical evolution became a theory. So essentially, Miller had this question: can complex organic compounds be made from simple molecules present in early Earth? So let's talk a little bit about how Miller set up his experiment to see if this hypothesis is correct. There's a picture of your book, and it's going to be a little bit more complicated than what I'm going to draw, but I'm just going to draw the basics of it. So let me explain what I drew here. This part of the apparatus is going to represent a mini atmosphere. And this part down here is going to represent a mini ocean. In the mini atmosphere, he's going to put 3 chemicals present in early Earth: ammonia, hydrogen, and methane. I don't remember if I said it, but there is water in the mini ocean, which would make sense, right? He then heated the mini ocean, the water. And what happened then was vapors would form as this boiled, and the vapor would travel to the mini atmosphere where these 3 early gases were present. He then had something we'll call a condenser here, and that would help condense these chemicals into the water droplets and hopefully maybe some kind of reaction, and then they would filter down in here back into the boiling water. He could then draw out samples from the boiling water and test them to see if any organic compounds had formed. Now, when he did the experiment this way, he didn't see any complex organic compounds forming. For the most part, these chemicals are quite stable, and so they didn't really form anything, they didn't break down, nothing happened, even though they were in boiling water. So he added something to his experiment. He added, essentially, lightning. I'm going to draw them like this. He put 2 electrodes here, and the electrodes simulated lightning. And then he redid the experiment. And at 1 day, the water here turned pink. Then at 1 week, the water turned red. When he tested this water, what he discovered was quite amazing. He discovered, I'm going to draw it over here because this is where I have the most space, compounds like hydrogen cyanide and formaldehyde. Now, while these chemicals turn out to be poisonous to us today, early on, these were probably pretty important because these are highly reactive compounds, and they could promote the formation of larger molecules. In fact, Miller even observed the formation of amino acids in this red liquid. These data from Miller supported the idea that the simple compounds present in early Earth with the right stimulus, say like lightning, could react, and in the boiling water even react more to produce larger compounds that could ultimately be responsible for forming life. So Miller's experiment and several other experiments helped build up evidence to support the theory of chemical evolution. Okay, let's go ahead and move on to the second objective, and that is that RNA was the first self-replicating system. So to introduce this topic, let's think about what we've talked about a little bit ago, called the central dogma, where we have DNA that is converted into RNA by a process called transcription. You won't need to know that right now. And then how RNA is then translated into a protein. And this idea of the central dogma here is very well established. However, it would be unreasonable to think that during the course of evolution, all 3 of these macromolecules, DNA, RNA, and protein, appeared at the same time. Rather, it seems likely that one of them came first. So the question now is, which came first? It seems, based upon some of the characteristics of RNA, that RNA is a good candidate as the macromolecule that appeared first. And 2 features of RNA that makes it a likely candidate is that RNA can store genetic information. Now, in eukaryotes and in bacteria and archaea, genetic information is stored in DNA. But even today we know that there are some viruses that store genetic information in the form of RNA. In fact, COVID-19 is an RNA virus. So we know RNA has this ability. So maybe it had that ability billions of years ago. The second is that it was discovered that RNA can serve as a catalyst. A catalyst is something that speeds up chemical reactions. When RNA serves as a catalyst, we call it a special kind of RNA called a ribozyme. Today we know that most enzymes are made up of proteins, but RNA can function as an enzyme, so we call it a ribozyme. And even to this day, our cells rely upon RNA catalysts, these ribozymes. Without them, we wouldn't be able to survive. So they still exist today and they're still important. So DNA and proteins would not have necessarily been good choices as the first macromolecule in this system here. And the reason for that is that DNA, while it has great ability to store genetic information, there's no evidence at all that it could be an enzyme. Likewise, proteins, they're extremely efficient enzymes, but there's no way they could store genetic information. So because of that, first idea was that RNA had to have been this first macromolecule. So this idea is known as the RNA world hypothesis. And essentially, this hypothesis states that during the evolution of life, RNA stored genetic information and catalyzed its own replication. So we might think of RNA doing something like this when it was the sole macromolecule, where you have this RNA molecule here and it stored genetic information in it along the nucleotides in the linear array here. It would then catalyze a new strand of RNA. So this was the original, what we'll call a template strand. I'll just put a T there. But it would then create a new strand. Now I'm just going to put N for new. And then they would separate. And you would then have two independent molecules that could then go on and catalyze a new strand once again. And I'll just put this one in red to show it as the new strand. And it would keep doing this over and over again. So one of the problems with RNA acting as a ribozyme to catalyze its own replication is that ribozymes are less efficient. And we predict would be error prone. So there might have been a greater ability to generate mutations as it was self-replicating. Now maybe this was good because maybe it helped speed up evolution a little bit to create some diversity within the RNA world. As I said on the last whiteboard, remember that at some point proteins and DNA came along, which then replaced some of the functions of RNA. That is, the functions of storing genetic information and enzymatic functions. However, RNA, as you know, still exists and still serves a very important role in our cells. And as I said, in the case of viruses, it can still serve as a source of genetic information. All right, let's move to our third and final objective for today's video podcast. And this is going to center around the first cell. And for this discussion, let's draw a structure called a phospholipid. First, let's draw a part of the phospholipid called these fatty acid chains here. I'm just going to put Fa for fatty acid. Then attached to the fatty acid chains is a molecule called glycerol. And at the top is a phosphate group. Some key features of this molecule. The first is that the tail has a property that we call hydrophobic. And this essentially means that these kinds of structures, hydrophobic structures, avoid water. The word means water fearing. So they try to get away from water. Up here, the phosphate has a charge on it, a positive charge. And so it has this property that we call hydrophilic, which literally means water loving. So it wants to associate with water. So you're not going to have to draw this on an exam or on a quiz, but you should be able to recognize the parts of it. You should recognize that this is the fatty acid tails down here and that they're hydrophobic. You should recognize that this structure here between the fatty acids and the phosphate is the glycerol molecule. And then that the phosphate group at the top is hydrophilic. And you should know the definitions of these two. Hydrophilic associates with water, hydrophobic tries to get away from water. So let me throw one more definition out to you. And that is when a molecule has both hydrophilic and hydrophobic properties, we say it is amphipathic. Just meaning it has both properties. The amphipathic nature of a phospholipid is extremely important in forming a vesicle. Because when you get a bunch of these phospholipids and you put them in water, the hydrophobic portion immediately stick together to try to get away from the water, and the phosphate portions of them interact with water. And in doing so, we end up forming a structure like this. Where the fatty acid tails are in the middle here, and the phosphate groups are on the outside, where all the water is. And what happens is as more phospholipids are added, it encircles itself. And it forms what we call a phospholipid bilayer. And I'm going to attempt to draw what a phospholipid bilayer is, but with much less detail, because I want you to see how it forms what we would call the cell. So this first green circle I'm drawing is this top layer. And then this bottom layer is going to be here. And I'm going to draw it really close to it because it needs to represent those phospholipids interacting with each other. And I'm not going to draw the glycerol, but it's there. I don't want to make the picture more complicated than it needs to be. I'm just going to draw the phosphates on the outside. And I'm just going to do that by drawing a lot of pluses here because it's positively charged, and that's what's going to be interacting with the water through hydrogen bonding. And now what we have here is a structure where the hydrophobic portion is interacting with each other. The hydrophilic portion is on the outside interacting with all the water outside of this vesicle, plus all the water inside of this vesicle. And this allows it to remain quite stable. I'm going to erase this part here just so I have a little bit more room. So what we have here is something we would call a liposome. So we have this nice lipid membrane around here with water on the outside, water on the inside. Now, inside of here with the water, when it was forming, it's possible that it captured inside of it an RNA molecule, one of these RNA molecules that has the ability to self-replicate. And now you have, with the new RNA molecule in it, what we would envision to be the first cell. Now the ability to replicate the cell, that is to copy the cell, and to do all the many functions that modern day bacteria, archaea, and eukaryotes are able to do, would be a very slow process, as we talked about at the very beginning of this podcast about the evolution of life on Earth. But this marked a very important event where we had genetic material protected inside of a lipid membrane bound structure. So to kind of take a snapshot at the big picture here, where we've been so far this semester, we started off talking about what is life. We talked about what it means to be alive, the characteristics of life, and all that good stuff. And then we talked about what is life made of. We talked about the formation of chemicals, larger chemicals, macromolecules. And today what we focused on is how did life start? And then where we're going to go next is how did we generate all the diversity we see on Earth? And that's where we begin to talk a little bit more, quite a bit more, about evolution. So the next podcast and next day of class, we're going to focus on introducing you to evolution. So as a quick summary, we began this podcast talking about the evolution of life on Earth. We then spent quite a bit of time talking about chemical evolution. We talked about how we could take simple chemicals, bring them together to form more complex and even more complex compounds. And we talked about Miller's experiment that showed that chemical evolution could have happened. And again, this is known as a theory. And then we talked about RNA as the first self-replicating system. And we talked about how RNA has the features to have allowed it to come before DNA or proteins. And then how RNA can serve as an enzyme by functioning as a ribozyme. And then we talked about the first cell. We talked about the structures of a phospholipid. We talked about its features of having hydrophobic and hydrophilic portions and how that helped form a membrane, and then how that membrane, if it formed around one of these self-replicating RNA molecules, that now you had a vesicle, so to speak, that could protect that genetic information. All right. I think that ends today's video podcast, and if you have any questions at all, make sure you come talk to me. If not, I will see you in class. Bye for now.